Hello, everyone. Welcome to the WebEVTalk series. So today is the 10th August 2021, both in Europe and in Australia. And I'm excited to have a young investigator with us, Anna Antoniou from Germany. So she's currently a research fellow at the University Hospital Bonn in Germany, and also at German Center for Neurodegenerative Diseases. So Anna did her PhD in Bristol, UK, where she worked on the molecular mechanism of synaptic plasticity in neurons. She then moved to Germany, where she received postdoctoral fellowships to investigate microRNA production during neuronal development. She is currently working at the University Hospital Bonn and Associated Institute of Neurodegenerative Diseases, working as a primary investigator. She's focusing on the regulation of dendrite and synapse maturation by extracellular microRNA. So today, she's going to share her project with us, which is neuronal extracellular vesicles mediated BDNF-dependent dendritogenesis and synapse maturation via microRNA. So before we start, I'd just like to thank Horiba Scientific, who is our sponsor for this session today. And if you interested to support WebEVTalk, you can also do so through patreon.com and search for WebEVTalk.
Okay, great. Thank you, Carolina, for the invitation. Appreciate it. And thank you everyone for joining. And today I'm excited to show you my work on extracellular vesicles from neurons and how they are implicated in BDNF-dependent dendrite and synapse development. Just as to put everything in context, since I know some of you may not be from neuroscience, I would like to show you how diverse neurons can be or are in the brain. And here shown is just three types of main glutamatergic neurons of the hippocampus, where you have dendritic projections that are pretty complex in structure, emanating from the cell soma. And in these cases, the axon would be projecting downwards, forming long axonal tracts. And this morphology is known to be optimized or related to the function of neurons, which is essentially to collect information in terms of synaptic inputs, and then make a decision on whether to fire down the axon. So dendrite morphology, it is known that composition and morphology of these dendrites is important in these functions. And as you probably know, dendritic inputs are formed via synapses, which are composed of presynaptic axon terminals, which release neurotransmitter following an action potential, and postsynaptic terminals or dendritic spines or on dendrites. And synaptic plasticity is referred is a term used to referring to the change in the strength of these connections upon experience-dependent or other forms of neuronal activity. And this can occur via various mechanisms and various pathways, including BDNF, which has a major role in strengthening these synapses, either via actin regulatory pathways or local protein synthesis. So we're talking about the hippocampus or hippocampal neurons, as I will explain. And it is known that the hippocampus is an important region in the brain for spatial memory as well as mood regulation. And we don't know exactly how memory is consolidated, but many very good theories have been proposed, which are also based on recent advances in neuroscience. And one of these, as shown here, is that upon learning, you have the formation of new synapses, or the strengthening of existing synapses, shown here by dendritic trees, which are at later time points, for instance, between 12 and even longer hours. These synapses are either eliminated, further strengthened, or there's formation of new synapses. And in cases of developmental stages or environmental enrichment, so if you place yourself in a new environment, this process is significantly enhanced, was more pronounced. And this is this is thought to be a molecular basis of learning and memory. So why we choose BDNF? Well, it's a very interesting molecule because it is a growth factor, it's a major growth factor in the brain, and it's acting also as a neuromodulator as well as a neuromediator. This essentially means that BDNF can itself mediate plasticity by causing long-term changes in synapses. But it can also mediate plasticity for instance, by regulating secretion of neurotransmitter and the activity of neurotransmitter receptors such as glutamate receptors, AMPA and NMDA. So it's some details about BDNF. It's secreted by both pre and postsynaptic synapses. It's extensively regulated and in terms of splicing and gene expression. It's very highly expressed in the hippocampus, and signaling by BDNF and binding to its receptor is thought to be highly localized. So BDNF doesn't diffuse out into the extracellular medium, at least not longer than 4 microns. In addition, it regulates local mRNA translation at dendrites and axons, which I will touch upon later. And decreased BDNF signaling is observed in many neuropsychiatric disorders, in particular depression, schizophrenia, and as well as dementia, where you have consistently low amounts of BDNF. So our question was, are EVs implicated in BDNF-dependent structural changes, as they have the potential to cross synapses? So for this, we started off by isolating neuronal EVs, of course, and we used kind of a standard method of separation using ultracentrifugation. And we first characterized these EVs by applying the exosome inhibitor GW4869, where we saw about 40% decrease in the amount of small extracellular vesicles isolated by 100,000 G. And we didn't see any significant changes in sort of medium-sized vesicles isolated by 10,000 G. So another aspect of neurons, because we're using neurons in culture, is whether these EVs are coming from neurons or other cells that are commonly present in cultures. So here we treated neuronal cultures with antimitotics, RXE, and we processed our pellets with using NTA, as here. And we didn't see absolutely no changes in the curve and concentration or size distribution of these particles. In addition, we didn't see so many glial cells in our cultures, which is reassuring that we have mostly neurons in our cultures, which are shown here by staining with MOP2, whereas astrocytes are stained with GFAP. So we also used scanning electron transmission electron microscopy to look at our EVs, where we saw nice circles as shown here, which were about 100 to 200 nanometers wide. And BDNF stimulated EVs from cortical neurons looked pretty much the same. So there was no big changes in particle size or the heterogeneity or the amount of particles in our suspensions. We further validated that BDNF doesn't really affect neuronal secretion per se in these conditions using Western blot, where we blotted for ALEX, THG, and flotilin, as shown here. or with NTA, where we processed pre-cleared supernatants. And again, we didn't see any significant changes in particle concentration. So our main question is, because BDNF is known to have a role of in the dendrite morphology, where it increases the complexity of dendrites. So we wanted to see, do BDNF EVs propagate this effect by BDNF. So here we treated neurons with either control or BDNF for 20 minutes, washed off the treatment, collected small vesicles by 100,000 G centrifugation, and then applied them on recipient hippocampal neurons, and looked at dendrite morphology around 72 hours later. And remarkably, we saw that incubation of BDNF stimulated but not control stimulated EVs increased the complexity of these dendrites. And this we quantified by shell analysis, which is shown down here, whereby we counted the number of times these dendrites cross these concentric circles. And for example, as you see here, number 1 is the first circle, number 12 is the last one. So we see, at least compared to control EVs with increasing concentrations, that you see an increase in dendrite complexity. That is not strictly dose dependent, but is also not decreasing either. So this was quite interesting to us. And then the first question was, okay, is are EVs kind of carrying BDNF itself and then transmitting it to extracellular regions, as was shown for a number of other proteins. So we first plotted for BDNF. So we did the same thing. We treated neurons with BDNF or recombinant BDNF, which is MIG tagged, and then we isolated cell lysates or SEVs. And then we check to see whether we see any BDNF signal or MIG signal. And as you can see here, there's little to no, absolutely no amounts of exogenous BDNF. In addition, BDNF EVs, in contrast to BDNF itself, did not activate signaling cascades downstream the TrkB receptor, namely the MAP-ERK kinase, which is strongly induced by BDNF, as shown here, for up to 3 hours, but not by addition of either control or BDNF EVs. So there has to be something else that's mediating this strong effect on dendrite morphology. We turn to microRNAs because, as I said, neurons have very diverse morphologies, with sites that are very far away from the soma and major organelles. They are important in regulating local translation at these sites, which are far away from the soma. And this was shown to be important for many aspects of dendrite and axon development, as spine maturation and plasticity. And we also know from previous work from my previous postdoc that these microRNAs are produced at these sites, dendrites. So we know from other research from the Schumann lab, for example, that we have this we have main organelles and ribosomes at these dendritic branches, which are close to synapses. And my previous work here showed that BDNF can influence the production of microRNAs, such as miR16 in this example, by redistributing the factors that produce microRNAs at these sites. So this could be a way of producing locally microRNAs in these arbors, and thereby regulating morphology of these sites. So we went ahead and performed small RNA sequencing of control or BDNF stimulated cell lysates and EVs. And as you see here with principal component analysis, there was changes in microRNA abundance in these samples. However, these changes were not at all correlated between small EVs or cell lysates, which means that the changes that we see in these EVs are not simply reflecting changes that we see in the cell lysates. And this you can see also here in this heat map of the top significantly changing microRNAs in the EVs. So from this list, we took the 3 most upregulated microRNAs, miR690, 132, and 218, and then we validated these microRNAs since they passed the statistically passed multiple comparison analysis. So as you see here, miR132, for example, is induced in cell lysis, which is previously known by BDNF, and as well as in EVs. However, this is blocked by inhibition of BDNF signaling by this drug UO126, which blocks MAP phosphorylation. We saw for 218, we saw the same in EVs, although there was no change in cell lysates, as I mentioned previously, and the same was for 690. In addition, we wanted to see whether these microRNAs or these changes are in vesicular fractions. So we used size exclusion chromatography, where we saw that indeed we see an increase in these fractions corresponding to vesicular fractions in SEC, but there was no changes or very variable changes in later fractions that are corresponding to protein, most likely. And as a note, we did see microRNAs in these non-vesicular fractions. However, we think that is somehow not really regulated. So furthermore, when we blocked exosome secretion using GW4869, we saw a decrease or depletion of these microRNAs 132, 218, and 181, but not 690 in small EVs. But no big changes in cell lysates, suggesting that at least 2 out of 3 of our regulated microRNAs are most likely associated with exosomes. So what are these microRNAs? So we know that, for example, 132 and 218 do play a role in many processes in neurons. So we looked at predicted targets, and we found that a lot of these targets for these 3 microRNAs are common. And when we performed gene ontology analysis, we were kind of happy to see that these targets are predicted to be implicated in nervous system development. And if you notice here, they are localized in postsynaptic density, or which is basically a synapse, postsynaptic synapse. And it's also interesting to see that some other things came up, like positive regulation of spine development, dendrite extension, and we also saw a lot of factors that could be involved in regulation of transcription, which may explain actually these large changes in dendrite morphology that we're observing. So this is all nice, but are EVs taken up by our neurons in these conditions? It was previously shown, but we wanted to check in our system. So we used this lipid binding dye previously called membrite, but sold as lipilide, and which has high signal to noise ratio, as shown by this publication. And was previously used also for EVs by the Goetz lab. So we validated that when we take EVs and label EVs from just cell culture media without cells, we don't have any signal, which was nice. Whereas with EVs, we see a nice strong signal in neuronal cell bodies and dendrites. Also, when we this is shown here as a neuron outline, as shown here. When we pre-treated neurons recipient neurons with dinosaur, which blocks dynamine-dependent endocytosis, we basically saw no signal in both control EV and BDNF-EV condition, which suggests that the majority of these EVs are probably endocytosed in our case. We went a little bit further and we did some super resolution imaging, super resolution-like imaging, where we saw that a lot of these puncta of lipid EVs are sort of colocalizing with late endosomal markers, which were previously proposed to be sites of EV content release. So this was nice, but does this mean that EV microRNAs are delivered functionally? So to answer this question, we use this sensor with N2A cells, which are neuroblastomal cell lines. And this sensor consists of two constructs, GFP and DS-RED, from the same plasmid and two different promoters. And a control sensor has a 3' UTR on the DS-RED, and we cloned in two miR-218 binding sites at the end of the DS-RED only. So when miR-218 binds to this construct, we would see reduced amounts of DS-RED expression, but GFP should stay the same. So we transfected our N2A cells with this construct, control or miR-218 sensor. And as you see here, in the absence of EVs, you see a decrease in the ratio of DS-RED to GFP, which indicates endogenous miR-218 activity. And addition of EVs after 24 hours, but not after 4 hours, decreased this even further by about 50%, suggesting that EV miR-218 is probably functional. In addition, we perform experiments to block the activity of endogenous microRNA using antisense LNAs. And as you see here, in the absence of EVs, this construct is de-repressed, so there's increased amounts of DS-RED expression, but this is partially rescued by addition of EVs. So we went back to our dendritogenesis assay, and then we asked whether these microRNAs are implicated in BDNF-dependent dendritogenesis. So here we blocked endogenous microRNAs using this antisense LNAs. And interestingly, we found that blocking of these 2 or 3 microRNA combinations is sufficient to completely abolish BDNF-dependent dendritogenesis. In addition, if we supplement in these conditions with BDNF-EVs, we don't see a decrease in dendritogenesis as shown here. As I said before, we know that some of our targets were predicted to be on postsynaptic sites and synapse maturation, and BDNF is known to have an effect on synapse maturation as well. So we wanted to see whether BDNF is actually, whether EVs have a functional role in this process as well. So we transfected neurons with DS-RED as a morphological marker, and we performed the same experiment, but then we stained for synapses using the postsynaptic marker PSD95 and the presynaptic marker synaptophysin to label synapses. When we blocked these 3 microRNAs, we didn't see a change in the number of spines as shown here. However, we did see that blocking of these microRNAs blocks the BDNF-dependent increase in the number of PSD and synaptophysin positive spines, which indicates it blocks synapse maturation. We again did some rescue experiments where we saw that when we applied BDNF but not control EVs, this effect was rescued. So as you see here, blocking of these microRNAs upon control EV incubation, reduces the number of PSD and synaptophysin positive spines, but this is reversed when we add BDNF-EV. So this suggested to us that BDNF-EVs may also be promoting synapse maturation. We went a little bit further to see whether we can see this also on another level where we performed staining with PSD and synaptophysin again, and we investigated colocalization of these two markers, which would again indicate synapse formation. And here in both cases, when we applied control, so BDNF or BDNF-EVs, we see an increase in colocalization, as shown by the number of colocalized voxels or puncta in Imaris, and in Manders colocalization. Manders overlap between these two proteins. We wanted to see whether this was specific to excitatory synapses, so we did the same experiments using inhibitory synaptic markers, VGAT and gefirin, and where we saw no changes either with BDNF stimulation or with BDNF-EVs. In addition, we wanted to see where this change was coming from, and since we didn't see any changes in the number of spines, as shown previously, we wondered whether this is coming from presynaptic sites. So here we stained neurons with MAP2, shown here in blue, which stains neural dendrites, and with synaptophysin, which, as I said, is a presynaptic marker. And when we add BDNF-EVs, as you can see very clearly here, we see an increase in the number of structures that are touching dendrites. And this was nicely reversed when we took EVs from GW4869 treated neurons, suggesting that exosomes are probably important in this process as well. In addition, this was similarly the case when we did the same experiment using VGLUT1, which is an excitatory presynaptic marker. In both of these cases, we saw an increase in the number of these structures, but not in the mean intensity of these structures, suggesting that it is not due to an increase in the expression of these proteins. Finally, we wanted to see whether these changes translated to functional changes, as you can have formation of synapses, but no without any changes in activity. So we plated neurons onto multi-well electrode arrays consisting of 16 of these electrodes, as shown here. And using these multi-well plates with the electrodes, we can measure responses from these neurons in culture. So as neurons mature, there's more synchronized network activity, and this is what we can measure here, as shown from these raw plots. From these raw plots, the software there then sort of isolates or plots again individual spikes. So this is measurement of neuronal activity. And these spikes organize themselves into bursts, which is defined as a specific number of spikes in a temporal window within each other. So this is an indication of organized activity and neuronal maturation. So we went ahead and added control and BDNF-EVs for 24 hours, and we measured responses. And here we see that BDNF-EVs increase the firing rate, so the number of spikes overall. And this was also the case for the number of spikes in the bursts, as well as the duration of these bursts. However, they didn't have an effect on the frequency of the bursts overall, and the number of active electrodes in our cultures was comparatively the same. Finally, we wanted to see whether this is kind of a functional aspect. So when we applied BDNF on its own and then applied BDNF in the presence of this inhibitor. And we saw here again that application of GW4869 blocks or partially blocks the increase in neuronal firing rate following BDNF application. On contrast, and rather interestingly, there's no change with BDNF on its own on the number of spikes per burst or the burst duration. And again, GW4869 has no effect on this either. So we think this may be a specific effect of BDNF-EVs in inducing presynaptic clustering, which would lead then to increased firing by these neurons. So this is ending here, and in summary, we showed that when we BDNF when there's localized signaling between BDNF and TrkB at these sites, we think that there is a sort of localized secretion of these BDNF-EVs and upregulation of certain growth-promoting microRNAs. And the release of these EVs and microRNAs potentially, as we showed, increases the number of dendrites and dendrite complexity, and as well as the presynaptic clustering at these sites. And this could be an effect, a localized effect on these dendrites, but in general, it functions to increase dendritogenesis, synapse maturation, and excitability. So there's a lot of questions left. As I said, we think it's quite an interesting pathway. It's the first example that we know of in neurons that we see this transfer of microRNAs between neurons. It's been shown between different other types of cells in the brain. So we want to see, we're working now on the functional targets of these microRNAs. And we want to also examine the spatial and temporal dynamics of EV release in neurons, as we think this will be quite an interesting thing to look at, as well as the spatial and temporal dynamics of EV uptake. Answering these questions will give us a better indication of how these EVs may contribute to BDNF-dependent plasticity. And whether this is sort of a general feature of neuronal EVs based on their ability to travel distances. And finally, of course, because BDNF is a very interesting molecule, in particular related to hippocampus and the function of the hippocampus, could this be in the future important for diagnosis or treatment of BDNF-related disorders. And with this, I would like to go to my acknowledgement slide. So I'm part of the host group of Professor Anja Schneider, and I would like to specifically thank Loic and Julia, which are two talented technicians which helped with the project, and our collaborators, especially Professor Fischer and his postdoc Lalit for small RNA sequencing experiments, and Professor Schoch and Silvia Casas-Cunileno for their help with multi-electrode arrays. For this project, I also received internal funding by the UKB, which was staff and material funding, and for my previous funding, fellowships for my previous project shown here. And thank you for your attention. I'm happy to answer questions.
Okay, thank you so much, Anna. Let's just close the sharing first. Okay. Yeah, thanks so much for sharing your data. And I've got a few questions here. So I was wondering whether you also consider other cargoes of the vesicles, like the proteins and and the lipid maybe. There is a lot of debate about whether microRNA in EVs are really functional or not. And I was just wondering whether you look also into the other types of cargo as well.
Yeah, that's of course an interesting, an important question, I would say. So we did look at some changes in the protein composition, but we didn't see anything striking using mass spectrometry. So we don't, our data doesn't exclude other factors, of course, but we think that based on our results, that microRNAs would have a significant role here. And I think the point is also maybe it's depending on the donor cell, the recipient cell, whether microRNAs are indeed functional in EVs.
So that was using mouse brain, is it?
Mm-hmm.
Yeah. Okay. So when you're talking about the changes in the morphology, would that be also possible due to the growth of neurons? Or is it like totally like a branching or something like that? Can you tell us a little bit more?
Yes, so we do have neurons in a sort of in a state where they're developing. So they're developed, but they're further developing. So for us, this is definitely implicated in developmental processes. But it's also known that in adults, these changes do occur, although to a lesser extent. So for us, it's kind of, it's mainly based on development, but also on morphological changes as a matter of proof of principle, I would say.
Very interesting. So there's a question from Thomas. Thomas, could you turn on your camera and ask your question, please?
Okay, hello, good morning. Thank you much for this talk, very nice talk. Thank you, appreciate it. I was just wondering, can you test this also in vivo or ex vivo slices, for example, this BDNF effect?
We want to do that, so it would just, it's a little bit difficult for us to find a way to do it in vivo, which is sort of specific. So we see that it is indeed in neuronal EVs. I think there are tools coming up for these approaches, but yeah, how we can do this in a sort of way that's endogenous, let's say, is a little bit tricky. I think ex vivo, it would be a very good option, and we're looking into these options as well. Yeah. But then it's again adding EVs on top instead of sort of tagging these EVs to know that they're coming from neurons. I think it's very tricky in this field in general.
Sure, understand. Thank you.
Christy, please go ahead.
Oh, sorry, I'm on mute. Hello, amazing presentation, by the way. My question was just regarding the role of BDNF in neurodegenerative diseases like multiple sclerosis, where there's the demyelination.
I'm not 100% sure, but it is likely because BDNF is also involved in oligodendrocyte processes as well. It is in general decreased in dementia, or in not specifically in dementia, but also age-related disorders. But I wouldn't, I'm not sure exactly how it's implicated in demyelination.
Okay, great.
Thank you.
So Anna, with the BDNF itself, does it actually being secreted, or you know, maybe as a free protein, or is it a totally endogenous like protein?
It's secreted. It's a secretory protein, and it's secreted mainly from presynaptic terminals, but it's also secreted from postsynaptic terminals and has an autocrine signaling. However, the interesting thing is that it doesn't diffuse very far, so it's only diffusing up to 4 microns away. So an advantage of the EVs could be that they extend the signaling a little bit further to nearby dendritic regions. So it's a very sticky and regulated protein.
Yeah, but have you check whether it's actually also in EVs or not?
Yes, we checked for that, but we didn't find that. Thank you.
I think that's great. I mean, because that's more convincing in terms of this is the EVs that is playing a role that through that processes rather than potentially a mixture of BDNF and the vesicles. So I think that's really cool.
Yeah, so we also looked at, because BDNF has a lot of effects on also gene expression, we also looked if our recipient neurons have changes, the same changes in gene expression, and we didn't see that at all. So we really do think it's a specific process, and we're just trying to figure out what the specific role of these EVs are answering this process.
So you're talking about increase the firing as well, isn't it? So would that be possible that it's involved in Alzheimer or Parkinson or other neurodegenerative diseases?
Yeah, it's in terms of firing, I think it's possible, although neurodegenerative diseases have a lot of different factors playing in, also involving other cells. So it's a little bit hard to kind of pinpoint what the role of each is. But definitely, since EVs are implicated in these disorders, I could imagine that there is definitely a role of this pathway in this process.
Yeah. Okay. Thanks so much. I think I satisfied my questions. I wonder if any more questions from the participants before I close the sessions. Last questions? No? Okay, with that, I'd like to thank Anna. Thanks so much again for sharing your data and a very cool and neat experiments you've done and address all the characterization and clear isolation methodology. So thanks so much for sharing that with us.